Before we start, I'd like to acknowledge the Aboriginal and Torres Strait Islander, traditional custodians of the land, the Wurundjeri people of the Kulin Nation. We pay respect to their elders, past, present, and emerging. We acknowledge and respect that Aboriginal and Torres Strait Islander people have always used resources from the land and waters for nourishment, medicine, and healing. Now I'd like to introduce our speaker for today, Professor Phil Hodgkin. Phil is the current joint head of the Immunology division here at WEHI. Before working here, Phil completed his PhD at the John Curtin School in Canberra. After this, he worked as a postdoc researcher in California. He is highly regarded internationally as his discoveries in T and B cell biology have contributed to original theoretical ideas for the field. His lab focuses on understanding how immune cells process signals to regulate proliferation, survival, and differentiation. He uses experimental findings to build functional models to predict how the immune system will behave and how to optimally target drug therapies to individuals. Please join me in welcoming our speaker for today, Professor Phil Hodgkin.
Thank you very much, Ash, and welcome everybody. I've titled this here, Immunity with an asterisk, and the asterisk you'll see why in a second, because I was asked to give an overview of immunity, and of course, you might think that that would be just the facts and some sort of textbook view. But I've taken the view that I'm speaking to a starting group of researchers. So this is Welcome to Immunology for those who are starting in research. So it'll be quite different. So instead of just the facts, you're being handed now a history of conceptual advances, a vast battery of data from experimental campaigns, and a collection of precious working models, the latest view of what we know. You're being asked to take this, nurture it, and to add to it and pass it on. So that's a different perspective from just reading the textbook, hopefully. And of course, I'd just like to add my congratulations for choosing the subject of immunology. I think it's at the forefront of science. It has been thrilling for me to be a part of it. And I think it offers great problems to solve. And importantly for you, there's a long way to go. There's many clear advances to come. I think you will love the scientific challenge, the piecing together of all the components, even though they're going to be overwhelmed by many of them. And that almost every new finding has medical consequences. So learn your craft, do it well, and you will have a great and enjoyable time. That's the summary of what I'm trying to achieve. So how do you go about being a researcher? So you need the tool of the trade here is generally called the scientific method. It too is evolving and learning to cope with biology, immunology, big data. It's changed. It's changing as we speak. But still importantly, hypothesis, the use of hypothesis in a precise manner is a great tool. And this is Karl Popper, who developed a whole scientific method based on attacking a hard problem by formulating a hypothesis in a way that you can prove it wrong. And you make advances by proving things wrong and making steps forward. And he taught us how to distinguish between powerful and weak hypotheses. And other aspects of the scientific method are that occasionally our perspective, we realise, is wrong. We get a revolution in the way we think about these things. And I'll give you an example of where this happened in immunology as well. So learn from the best, adopt their strategies. Now, so let's get into it. If you lift the lid of the immune system, what will you find? I'm going to do this now in one slide. This is what you find. A huge concatenation of lots of things all happening at once. In the human immune system, there's a lot going on. There's a lot of moving parts all jumbled together. So let's try to unravel it. And in the first instance, let's take a little bit of a historical perspective. And I like to do this because it lands us back in Melbourne, which I think you might find interesting as we're here in Melbourne. Okay. So we'll do this quickly. Typically, there's been some early consciousness that there's some levels of immunity. That's Thucydides, who wrote about protecting against plagues during the Peloponnesian Wars. But often we'll start here with Jenner and his idea that cowpox might protect an individual against smallpox. And we've just survived a pandemic. So if you haven't come across this story by now, the development of vaccines, so Pasteur extended the idea that lots of diseases arose from unique individual infectious organisms, the germ theory. And Pasteur realized if you could provide a weakened form of each germ, you might evoke some sort of immunity and offer protection. And so as a consequence of this simple insight, the development of vaccines has revolutionized public health, of course, including more recently the SARS-CoV-2 vaccines. But that doesn't tell us what's going on when you deliver those vaccines, what's going on under the hood. And for that, it's a long story. But I just wanted to give a couple of key points. So this is back in late 1800s, early 1900s, German scientist, microbiologist von Behring, showed that after immunizing guinea pigs, you could take the serum and transfer it to other guinea pigs and get protection. Or you could take the serum, put it in a test tube and kill diphtheria bacteria. So there was something produced as a consequence of being exposed to the infection. He called it antibody. And then the mystery was, well, how do you make antibodies against something specific? And around about in the 1920s, Karl Landsteiner, a chemist, made lots of chemical components that had never been seen on Earth before and showed that he could make antibodies to them. So that sort of eliminated the idea that you've got ready-made antibodies against any component. If they've never been seen by evolving organisms, how could that be? So for a long time, the dominant theory of how antibodies would be specific was provided this way. So just to summarize, this is championed by Linus Pauling. Linus Pauling was a very famous chemist. He made it onto a stamp. He won 2 Nobel Prizes. And his idea was that antibody would fold around something foreign we're going to call an antigen, and would be manufactured. So way down the complementary shape of the foreign material would be replicated in a complementary fashion in the antibody, and it would be manufactured in this sort of fold and release way. This idea was eviscerated by Macfarlane Burnet. Macfarlane Burnet was the director of the Walter Eliza Hall Institute here in Melbourne. And he did one of the very first simple experiments. After you immunize, he followed the appearance of antibody over time, and he generated this graph. This graph is a log scale here in the y-axis, so this straight line indicates exponential increases. Now, Burnet had a strong hypothesis. He didn't just say that the immunization should lead to an increase in antibody. Pauling's theory is right. It will increase in a linear fashion because it'll be produced in this linear way. So it should increase in a linear way. So the fact he saw an exponential allowed him to say that theory must be wrong. Now, he didn't know what the right theory was, and it took him 10 years to get to the right theory. But the point was, this is a powerful use of hypothesis. Make a strong prediction, not a weak one, that it just goes up. It goes up according to a certain pattern. So it took him 10 years to revise the knowledge of immunity and come up with a new theory. He was so uncertain about how it might be received by the scientific community that it published in a very obscure journal, effectively a newsletter of the Australian Society for Science. Here it is in 1957. It's 2 pages. The first page is all introduction, and in the second page here, many of the critical starting points for the modern science of immunology are flagged for the first time. So here's the idea. So the idea was that you anticipate by generating clones of cells with a randomly rearranged receptor, where each cell has a different random rearrangement. So it has the capacity to see something different. You generate millions of these cells in this random way. And then antigen, when it comes in, the foreign material, is sampled and tested against each of these cells such that when it finds one that there's complementarity in the receptor, those cells are stimulated to divide. Those cells are stimulated to divide, ultimately becoming antibody secreting. So they take their receptor, which is the antibody, and secrete it into the serum, and it circulates around the body. So within 10 years, there was general acceptance of this theory. The idea that one cell would make one antibody was confirmed a year later by Gus Nossel, shown here in this picture. And ultimately, the problem of specific antibody was acknowledged as being in the domain of cell population dynamics arising from a Darwinian-style mechanism of gene mutation and cell competition. So this was a great theory. It changed the paradigm, changed from thinking about the problem in terms of molecules to the problem of population dynamics. It gave it an axiomatic structure, and it allowed us to deduce the outcome and predict features of tolerance. So how do you tell the difference between self and non-self? So as you're generating those cells in the early stages, you delete those that are seeing self so that the ones that are left will only see something that's foreign. Explains immunity, because once you see something foreign, you evoke this program of proliferation and differentiation to become antibody-secreting cells. And it explains immunity, the memory of immunity, because having generated all those new cells, you're armed and ready to go, and you've got many more as a starting point for the next infection. So we still celebrate and remember this great contribution, but here's an editorial and the celebration of the 50th anniversary of that theory. Burnett's clonal selection theory not only had great implications for modern immunology, but was also monumental in spearheading immunology research in Australia. In 1957, he announced a switch from virology to immunology research at the Walter and Eliza Hall Institute, and was instrumental in attracting some of the great names in immunology to the Institute. What followed was a series of key discoveries that have given Australian immunology research a world-class reputation. And hopefully you will continue on building that reputation. Okay, so let's jump through quickly to now. How has it survived? How has the theory survived? Well, one of the great immunologists that was recruited to WEHI was Jacques Miller. Jacques Miller discovered the immunological role of the thymus, that in addition to those antibody-secreting cells, there's another class of lymphocyte that had to develop in the thymus. Jacques invented a way of removing the thymus from neonatal mice, and they developed all sorts of immunodeficiencies. He also was able to show that the B and T cells have to cooperate to make antibody. Sorry, I jumped there. The cells ultimately that make antibody are called B cells because they develop, as we'll see, in the bone marrow, or the bursa equivalent in the chicken or birds. And the T cells, because they develop in the thymus, are called T cells for clear reason, but they somehow cooperate to make antibody. So that beautiful, simple theory of Burnett has to be complicated, more complicated. So I'm going to quickly run through to today and show you some of the highlights of overview of what you need to know to understand the structure of the immune system. So T cells, we know they mature in the thymus and that they circulate as resting cells, just looking for trouble inside our cells. For example, they're looking for a virus-infected cell or a cancer cell. To process this trouble, the T cell requires a presenting cell. It requires information to be given to it, and it needs that for two reasons. The first is they can't see native antigen in the way that the B cells can. But it must be presented on a cell surface with a self molecule, a self marker molecule, the major histocompatibility molecule, MHC. And this was a discovery of Doherty and Zinkernagel, and many of you will be aware of Peter Doherty, who also lives in Melbourne. There's an institute with his name on it just down the road, and of course he played a very prominent role in defence of vaccination during the pandemic recently. Okay, but antigen alone is not enough to activate the T cell. They need additional signals, and these are numerous and ultimately affect the strength of the T cell response. A key presenting cell, a dendritic cell, accumulates antigen, presents it to the T cell. It in turn has these toll-like receptors, which I'll say more about a little bit later. And these toll-like receptors engage with features of a pathogen, like components of the pathogen, and provide additional signals that upregulate additional receptors in addition to the T cell receptor, co-stimulatory receptors, as shown here. For CD28 is one here. Okay. The other important point to make is that there's a whole entire class of immune decision making that Burnett missed, and we call this class, that the immune system has options that aren't all used. There's a matching of response to infection. So not all ways of fighting an infection equally work effectively. For example, there's multiple types of antibody as shown here, just illustrated here. So this antibody is best for attacking worms, and this one might be best for attacking viruses because they engage with the immune system in different ways. So learning the different classes of immune response and how they work is an important part of immune function. So the clonal selection theory has to be modified to include the regulation of class, and we know in some level this is due to the transfer of signals. Information about the type of bug is transferred to the lymphocytes, T and B cells, as they're developing. That helps them to make those decisions. The third point is that B cells develop in the bone marrow and they circulate as resting like the T cells, and they have activation criteria that also are kind of not simple. Antigen alone is not enough. There's two different usual classes of antigen activation for B cells. There's a T-independent, where the antigen, as shown here, the antigen is captured on the surface of the B cell. Antigen is captured on the surface of the B cell, and it's attached to some other component that engages with another receptor, like a toll-like receptor, and providing second signals to the B cell that fully activate the cell. There's also T-dependent activation, where the B cell captures an antigen with its receptor, pulls it inside, processes it, presents peptides with the MHC on its surface, like the dendritic cell did, but now the B cell's doing it. And it's presenting here to the T cell, and if the T cell is specific for that antigen, it responds by producing a reciprocal stimulation. So it gets activated, produces a protein called CD40 ligand onto the cell surface. CD40 ligand engages CD40, which is already expressed on the B cell, and that provides strong activating signals. The T cell during this process can also provide further information by providing cytokines that help direct what type of antibody the B cell should make. For example, interleukin-4, a cytokine, might help direct to the antibody type IgE, which helps fight parasites. Okay. Next little bit of an overview is that you need to know that B cells have a two-phase response. So typically, B and T lymphocytes are circulating around the blood and through the lymphatics and through the lymph nodes, which are stationed all around the body. In the lymph nodes, they occupy different sites. And when if they see antigen, if the B cell sees antigen in its site, it moves and looks for T cell help. If it finds it at the border between the T and the B cell areas and receives stimulation, they start proliferating, they start making antibody. They change the type of antibody and they start secreting it into the body. So this is the first phase, proliferation, choosing the type of antibody and secreting antibody. This is all the production of early antibody. The second arm, some of those selected cells enter what's called a germinal center. And in the germinal center, they're taking a longer term view. So now they're going to cyclically mutate their receptor and look for higher affinity versions and select them. So there's a process of cycles that select and improve and select and improve, such that the product of the germinal center is higher affinity antibodies, either a memory cell, which will circulate in a resting phase, or an antibody secreting cell with high affinity that now, unlike the early ones that were produced in that first phase, now they have a very long life. And they migrate to the bone marrow and sit there and make antibody for a long time. And we don't know why it is that in some infections like measles, they sit there for years and years and years and you're protected, whereas with coronaviruses, as we've seen, we often get very short-lived protection. And that's one of the mysteries that hopefully you'll be able to solve. Okay. So the other point to make is tolerance is ongoing after deletion, that T cells seeing self in the periphery. So there is clonal deletion when cells are developed, as envisaged by Burnett. But even after maturing and going to the periphery, there's ongoing selection processes. So T cells seeing self in the periphery out of the thymus can lead to death. So there's a sort of backup mechanism. And okay. So in this broad overview, I was just trying to make that that we've determined that T and B cells are complex signal processing cells that are constantly making decisions that affect cell tolerance, immunity, the class of immunity, and memory. And that this system already has revealed many secrets. But in doing so, I make the point that we've lost much of the simple elegance of clonal selection theory. We now have a theory that has lots of variations and caveats and empirical rules for roughly predicting the outcome of an intervention specific for individual cells and situations. But we're very poor at predicting how to control the immune system. We also have a massive number of components that we're not quite sure how they all fit together. And so we have a huge amount to learn and build on. There have been heroic attempts to maintain the lineage of the theory of clonal selection. And without going into detail, there's this beautiful theory of Charlie Janeway that innate sensing, those toll-like receptors are channeling information to the T cell arm. And here's another beautiful theory presented by Polly Matzinger, that we can categorize these threats and others as a filtering, that the immune response doesn't have to mount a campaign against everything that's foreign, only those that provide threats and potentially damage our bodies. But famously, and maybe you already have this view, and maybe those online already have the view, that immunology has a reputation for being incredibly complex. And so during the pandemic, this article came out in The Atlantic, "Immunology is where intuition goes to die," which is a bit sad. And it says it's too bad because we really need to understand how the immune system reacts to the coronavirus. So I'm going to spend the rest of my time trying to look for a way out of the confusion. And the way out, I think, emerges from studies of integration of all these signals, decision-making by T and B cells under controlled conditions, and seeing if we can generalize them to more complex situations. Can we find an easier way to interpret this very complex system? And I think the provisional answer is yes, and that we can turn the clonal selection theory from a collection of heuristic rules to a quantitative theory. And the key for this is what I'm going to call for this talk the adaptive immune sensor. And the idea is that underlying all T and B decisions based on receptors on their surface is a simple autonomous program that responds to signals in a quantitative manner, that yielding a continuum of outcomes that are not a series of rules for individual situations. That this sensor replaces two-signal theory, the idea that T cells need antigen and something else, something from the innate system, and explains peripheral tolerance decisions. We put all of these components as a simple quantitative outcome of the one sensing apparatus. And I want to just give a quick overview of that and then finish with an insight, try to use this as an insight to develop an even broader theory that might provide a way of putting all the pieces together. So here's the sensor. The idea is, and it arises from lots of experiments as I'll show you, but the idea is that the T and B cells can be thought of as having multiple timers. So the receptor stimulation comes in and programs timers that dictate the first timer, the blue one dictates the time it starts division and how much time it spends between each division. The second timer, the green one, determines how long you divide for before you naturally stop. It shuts everything down and you go back to being a resting cell. And the third one clears the cells and kills off and kills all the members of the family. So as a consequence of the action of the three timers, you end up with these quite symmetric trees where they proliferate, stop dividing, and then all die at the same time. And our insight into that this might be happening with T and B cells came from direct filming. And this was lots of campaigns initially led by Edwin Hawkins, who's now a lab head here. But following the cells, we could determine that they were behaving in this particular way. So here's an example of a T cell in a tiny well. It's just received a hit. It started proliferating. And you can see how all the progeny of this cell start dividing at the same time. And they divide synchronously. And they all do the same thing. They all divide 5 times, and then they all stop dividing and all start dying at the same time, the whole family. Now, you might think it's because they're all in the same well, but if you mix cells within wells, you can follow them through individually. All members of the same family do the same thing. If we look within families, we see lots of variation. Some divide a little bit and die after a long time. There's all sorts of variations, but within a family, there's strong familial behavior, despite rich variation between clones. And the total population response after we stimulate cells, they start dividing, they proliferate, they stop proliferating, they die over a long period of time. There's a classic looking immune response that we're replicating with this behavior. Is the total population the sum of all these unique but symmetric families? So our analysis identifies timers that work across generations. So somehow the timed information is unaffected by whether cells are dividing or not. If we slow down the divisions with drugs, they all stop dividing at the same time, not after the same generation count. So there it is illustrated again. The signal comes in, we program our timers, we dictate the rate and the generation of this of these times. Now, when we do that in a population, we see variation. So there's an average division time and an average cessation time and an average death time. But within clones, there's a variation. We can describe that variation, it turns out, by these distributions. So with a simple model where we have a distribution, a mean, we can replicate the outcome. So we've been working on mathematical descriptions of that in order for us to apply it to data to work out how signals are integrated. So the idea is we don't like filming cells because it's really hard and following them, but there's simpler ways of following the dynamics of population stimulation in controlled conditions. We can label the cells with a dye and track their division using flow cytometry. And when we do that, for example, here, this population response, cells proliferating, stopping, and dying, if we look at each individual time point, where the cells are in each generation, you can see average 2 here, 3 here, 4 here, 5. With time, they're moving through the generations like this. And we can take our model and work out the mean and variances of these distributions to fit the data so that these blue lines are actually fitted model data to the red actual data points. The other thing we've discovered by using the system is that it's immensely controllable in an amazing way, so that there are signals, for example, here, I've just replicated a receptor that can send a signal that just affects the division time. It might speed up division time, but not change the division, the green time or the red time, the time they stop or the time they die. Similarly, we can have receptors that send a signal that just change the division destiny, we call it the division destiny time, when they stop. or we can have a receptor that just changes the death time. These can be independent processes. Or we can have a compound receptor that does some mix of the two. So this receptor doesn't affect the division time, but affects the time to stop dividing and the time to die. And that the signal addition and population changes that we're seeing here, we call the cellular calculus. And that the effect of any signals in combination, drugs in combination, gene changes can be quantified by the impact on these timers. And we're doing lots of work in that direction in the lab. Here's an illustration of using of cell calculation. So this is a paper we published in 2014 showing that T cells can integrate co-stimulators in sequence. So the number of times they divide is a function of the strength of stimulation and the number of co-stimulators. We took that data and applied it to our timer model, which is a more sophisticated version, and we could show that this is T cell proliferation, cessation, and loss just with TCR engagement. If we add one signal, we get an increase to here. If we add another signal, we get an increase to here. If we add them both, we can do a simple addition of the distributions. And the brown data is what we get, and the dotted line is what we get by prediction of the addition of these lines. Let me just paraphrase that with a little simple cartoon. So with one co-stimulator, we're getting a shift in the green and the red like this. We put in a second co-stimulator, we get a shift in the green and the red like this. If we put them both in together, we just add the two shifts together and we get the consequence. And the important thing is that we're adding signals in time in a linear way, but the effect on their clonal expansion is geometric, because each time you get another division, you get a twofold expansion in cell number. So we're converting a series of linear inputs into a geometric output. So what's the molecular mechanism for cellular calculation? And this is the beautiful work of a member of the lab, Sue Heinzel, and I'm going to tell it to you very quickly. The number of divisions is governed by a division-independent timer mediated by the oncoprotein MYC. So it works like this. It started as a hypothesis, and we drilled down, and we're now confident it works in this way. So the signals come in, they motivate the production of this protein MYC, as shown here, or in the intensity of green here. And MYC ultimately acts like a timer that's transmitted. When it falls below a threshold, the cells stop dividing. So let's just have a little illustration of this. So here's the timer. So the cells get stimulated. There's a pulse of MYC like this, and while that's pulsing, the cells will be able to divide. If we provide a weaker stimulus, there's a weaker pulse. The cells don't divide as much. They fall below the threshold. If we provide a stronger stimulus, now the pulse lasts longer and the cells divide for more. So you can see that pulsing, the adding in of the signals, the multiple signals, is channeled through the level of MYC and the timing of this pulse. Here's an example of the raw data. So here we've taken T cells, stimulated them through the T cell receptor, and added co-stimulators at different strengths. And you can see the MYC pulse rises and falls. As we increase the stimulation strength, the MYC pulse takes longer and longer to fall below a threshold. Simultaneously, we can measure when cells on average stop dividing, and we can follow that through here. And down here is an expanded view of the middle panel showing that the level of MYC where the cells predict to stop dividing is a constant level, confirming that we're seeing a threshold behavior. Okay, so the threshold trigger controls division destiny in this very simple way. The work of Michelle Rule in the lab is to look for the other fates. Can we get to the bottom? And it turns out they work in a very similar way. The death is being controlled by a timer that works in a similar way through the production of anti-apoptotic BCL2-related family proteins. And that the control of the division time looks to be related in a different way, the threshold of reaching a threshold to initiate division, and those proteins appear to be cyclin Ds. So the combination of these critical proteins contribute to this burst of proliferation, achievement of cessation, and the leading to death through that's what's going on. So to summarize, the healthy immune response for T and B cells is controlled by default timers that use time pulses of fate-controlling proteins. And it's interesting that all those proteins appear to be oncoproteins. And there's a connection, therefore, to the development of, for example, lymphomas that we're also investigating. Now, the use of modelling highlights how multiple inputs into these timers can sum to create large net outcomes in response. That the timers sum co-stimulators and danger signals as provided by the toll-like receptors, for example, to enhance the outcome. That we don't have a series of decisions, as in, if I got this much, then I should do this. If I've got, if I add A, B, C, then I need to do Y. Rather than an all or none decision or binary decisions, we have a simple continuum. And we can think of it like we're adding signals to say, how confident are we that this thing that we're seeing is foreign? I've got a strong signal, another cell's telling me they're getting a signal, you're adding them all in, and that dictates the strength of the response. So the strength of the response the cells are processing all the time can be so weak that they just always die, and that would presumably the situation in the periphery where you're coming across self-antigen without much additional signaling. Whereas a strong stimulus leads to a rapid proliferation for over a long period of time, and eventually when they stop, there's a very slow loss of cells because the times to die are greatly prolonged. So there's lots to do in fleshing out this theory more. For example, we want to look at how do the cells, is there a cyclic process having returned, stopped dividing, returned to this resting state? We think this is now a memory state, and so we can reactivate and go through the process again, set the same program. In the germinal center, we see it as this same program, the same sensing use of timers cycling through and being corralled and captured in a certain way. So we imagine we can use the same theoretical concept to apply in multiple different complex scenarios. We need to work out how we allocate cells to different fates, like how many cells become memory, how do they become antibody secreting, how do they become the different types of T cells making different types of cytokines. All of that is yet to be done. And there will be lots of challenges ahead. But I wanted to finish with an attempt to put all of this into a dream of even more range. I thought we would have a go at the theory of everything immune, the TOEI. So let's jump back into the everything, everywhere, all at once, and realize that really the key to pulling apart a very complex system where lots of things are happening simultaneously is to think about time. And what I just showed you with the dynamics of proliferation is also use of time to pull things apart. But let's, the famous quote from this physicist, that time is what prevents everything from happening all at once. That's our guide here and our friend. So let's just take a step back, that every species has a story of progress that includes infection over and over, that natural selection will leave its mark, reuse and repurpose the tools at hand. As immune defense is essential to species survival, when we've looked, due to this tortuous history, 10 to 20% of genes appear to be immune-related in most species. So look and see the development of the patterns and parallels to get insights into how it's all fitting together. Now, the idea of using evolution to understand human immunity was actually has a great history. So this is Eli Metchnikoff in the late 1800s, who plunged a thorn into the starfish larva and showed that the hemocytes, lots of phagocytic cells, appeared around the thorn and started consuming bacteria. And he saw exactly the same behavior in human pus, where macrophages, similar to these hemocytes, would phagocytose bacteria. So drawing a direct link between the evolution of immunity and these more primitive organisms to ourselves. But I want to use toll as an exemplar of how to put all the pieces together, because I think it provides a very powerful perspective. Toll is a protein originally identified in Drosophila because a mutation would lead to infection, as shown here, with this poor fly has a fungal infection. Now, what toll does is it sits on the fat body cells and it gets ligated by another fly protein called spatule. And spatule is converted from an inactive to an active state by serine proteases that themselves are dependent and derived from infectious organisms. So the infectious organisms drive the production of this ligand, which stimulates toll, which stimulates the production of antimicrobial peptides in this sort of stimulus response. And you can see that there's an importance to match somehow the stimulus to the effective response. I should say too that toll was originally identified as a developmental effect in flies, so it's playing multiple roles, and that's a common process, common feature. Now, if we look at toll in vertebrates, they're also found, but with a twist. So now you find multiple tolls. This is in human, for example, but they are varied. So there's toll receptor 4, toll 2, toll 1, toll 6, toll 5. They're all shown here on the receptor, and you can see that they are recognizing different components of pathogens. So this one is seeing lipopolysaccharide, which is found in bacteria, triacyl lipopeptide, diacyl lipopeptide, flagellin, also bacteria, double-stranded RNA, single-stranded RNA, DNA. So you're under attack. The toll-like receptors have been evolved and found themselves in the germline to detect those invaders. Each of them is connected to an adaptive series of adaptive proteins and sends a signal that motivates an effector response. And the effector response, you can imagine, has been matched to the type of pathogen that you're detecting. Now, most mammals have a set of 10 to 13 toll-like receptors with different pathogen-associated patterns that they recognize and different responses that they've attached to them. So you can imagine they've all been collected during evolutionary campaigns and battles with pathogens. That evolution and selection has optimized the specificity and the matching of the bug to the output, to the type, the strength, and the persistence. And that after Charles Janeway, who proposed that type of innate sensing might channel into the activation of the adaptive arm of the immune response, the T and B cell arm. This turned out to be correct, and I gave you some examples of that. But that provides the detection of these pathogen-associated patterns, stimulates, for example, this dendritic cell, and upregulates proteins such as CD80 here, which provides a co-stimulatory input into the naive T cell. So there's an indirect way of attacking the pathogen, promoting the adaptive immune response. Over here, I've just tried to show you, make the point that this toll-like receptor behavior is just like playing Lego in the genes. So there's some aspect of a pathogen, so you need to find a molecule that detects it. And so you can duplicate the genes, mutate them, and look through evolution, through natural selection in the germline, that if you find a recognition and then hook it up, adapt it to the appropriate signal and the appropriate response so that you match this response to this pathogen, you can have a profound effect on immunity. And so by changing the pathogen, changing the receptor, changing the adapter, changing the signal, changing the response to match it is what's happening over and over and over again. This very simple canonical behavior. Okay. And of course, the other point I wanted to make, to link it back to the rest of the talk, that instead of thinking about the innate system being an essential trigger of the T and B cell response, I want to put in here the possibility that there's this immune sensor, that there's a continuum, the signals are added and they dictate the sensing and the proliferation and survival of the T cells and B cells as we've seen. Okay. But there's a twist again, and the twist comes from this eel-like creature, the lamprey, which is parasitic. It lives off fish. It's, you can see the amazing teeth attaching to swimming fish and parasitizes them. But it turns out that the lamprey is a jawless vertebrate. Early in the evolution, it precedes the jawed vertebrates. And through a series of studies, mainly from the lab of Max Cooper, it's been determined that the lamprey has an alternative way of doing the adaptive immune response, the determinate set of receptors. It uses toll-like receptors and not the antibody-style T-cell receptors or antibody molecules that are used by the vertebrates, the jawed vertebrates. But the amazing thing that this paper says, not only are they using the toll-like receptor as their foundation for an adaptive immune response, they have all the same features of the cellular response. So they have a B cell, they have a T cell, they have different classes of T cell. In other words, so I'll just read this. The immune systems of jawless and jawed vertebrates are representative species of the two lineages use different sets of genes and different combinatorial mechanisms to construct diverse antigen recognition receptors, but they share the same basic principle of differentiation along two distinct T cell-like and B cell-like lineages. These suggest that the basic genetic program regulating the developmental pathways of the three lymphocyte lineages must have already existed in a common ancestor for all vertebrates. And this constitutes a fundamental organizing principle for lymphocyte-based adaptive immune system. To paraphrase, instead of acting on adaptive responses indirectly, as in most vertebrates, in lamprey, the toll-like receptor sequences are used as the equivalent of the T cell receptor and B cell receptor that we use and are connected to T cell and B cell equivalent cellular programs. Thus, the cell programs are also primitive. So here we've taken a different strategy. So we've taken our Lego, we've either can use the antibody style genes that we use, or we can use the toll-like receptor genes as used by the lamprey, and presumably evolution could have tried other protein sequences for this trick. Which receptor to use to vary, which gene randomizing method will you use, and there's multiple of those. How do you connect it to the response? And now instead of having a simple response, make a peptide and attack the bug, now we're attaching it to a cellular program. And the cellular program I propose is equivalent to our immune sensors, allowing over evolutionary time the battle between infection and survival to optimize the features, the distinguishing between self and non-self, the strength of response, how strongly should you respond, and how many of the clonal army should you make in order to appropriately protect you, and how long should memory last after that. All of those processes are playing out over the evolutionary time to optimize the system so that it works appropriately. So to finish, we can see that over evolutionary time, the accumulation and optimization of immune strategies that are small in number but infinitely variable in detail. So that successful strategies accumulate and promote survival of the species, and hence each immune system will be a complex mix of many overlapping versions of these successes. Thus, while seemingly infinite in detail and complexity, perhaps one day they'll be interpreted with a single unified theory of immunity, such that where immunology is where intuition goes to die, we can knock that on the head and say that we can make it very intuitive. And once we reach that point, we can determine how the immune system goes wrong in some people and causes autoimmune diseases, allergy, asthma, weak responses to infections and cancers, and how it could be controlled by design. And to achieve that, I'm going to hand it over to you as a puzzle. And I hope I've acknowledged the people as I went along, and I think I will stop there. Thank you.
Thank you again, Phil, for an excellent talk about discoveries in immunology and the work of the Hodgkin lab. Now I'm going to hand it to the floor if anyone would like to ask some questions.
Chocolate. Reminder that there's chocolates.
Come over.
I also have lollies if you're lactose intolerant. Natural fruit company if your mom's made.
Thank you for this brilliant talk. I learned a lot. Like I have questions about the part of signaling proteins that actually control the clonal expansion cell death of the immune cell that you have mentioned. Like you have said that in a cell culture in vitro system where you put all those signal protein into the culture, and you will see these immune cell can precisely be regulated how much they divide and when they going through cell death. But I wonder in a in vivo system like in your body, there's a lot of cell clones, immune cell clones, and how our immune system actually make the decision of which cell clone should expand more and which should expand less, and which clone should be death sooner and which should be later. And once this decision is being made, how it is being actually carried out by our body. Thank you very much.
That's a great question. I think it encapsulates the next phase of the research program. I think there's multiple points to your question. One is, how do we deal with the complexity of signal integration in vivo, in complex scenarios? And of course, it's hard, but my point of view is that if we know what's happening in these controlled systems, we know what to look for in those more complex scenarios. My view is that even though when everything's all mixed up and happening together, that at its core, these same processes are operating, although we're not keeping track of all the signals in the same careful way. Having said that, of course, there's lots of selection going on. There's affinity space selection. And their cells are talking to each other, and talking to each other makes a difference. So if there's other cells responding at the same time, they're going to make cytokines that feed over into the other cells, and it all goes into the big mix. My guess is that we're going to deal with that by saying we're not going to be able to predict the fate of every individual cell in a lymph node as a consequence, but we can use, again, probabilities and assess the population dynamics reasonably accurately, even in vivo. And we've had some success doing that in our experimental systems, modeling the dynamics of T cell responses after infection in vivo. So it's a hard question. We think it's going to be possible, but it's clearly challenging.
Thanks, Phil. It was a really great seminar. I really liked how you were trying to summarize some aspects of the immune response in mathematical terms. I think it somehow makes it kind of easier to try to predict the outcomes. But sometimes, we all done a cytokine multiplex and an immune cell phenotyping, and we have all this data, and we're like, okay, but what does that actually mean? Do you see ourselves coming to a point where we're going to be able to predict that in maybe not a very simple mathematical formula, but with math, that we summarize which cytokines are increased or decreased, or which cell populations are increased or decreased, so we can predict immune cell outcomes, or like the outcome of the host in infection, or how good an immune system of someone is? Do you see that happening at some point?
Another great question, and the intersection of the types of approaches that I've described and the other approach where there's this massive data collection and now ability to look at single cells in enormous depth and seeing lots of heterogeneity, how does that intersect with what I'm seeing? My view is that they will come together, and that ultimately what you're seeing with the high-tech investigations at this sort of high-dimensional single-cell level is revealing the patterns that are emerging as a consequence of all these simple autonomous programs that are operating. And that if we know how they work, as we work it out from these simple systems, once we know that those patterns, we can use our math to deconvolve what's happening in that more complex situation. At least that's our dream, that will happen.
I'll ask a question. I want a chocolate.
Does Chris get a chocolate if he asks a question?
Yeah. I'm really interested in the mucosal interface and how you decide on whether to mount an immune response or not when you've got commensals living there in the gut, for example. So do you think that the principles that you're talking about with, this is mathematical principles occur on the organ level as well? So how do you decide if there's a commensal there whether to mount an immune response? Do you have to get multiple additive effects to allow for that immune response to mount? How do you think it operates? Does it operate in a similar way at the cellular level that you talk about?
Yeah, another great question. Another question toward, the complex scenario. I think in mucosal systems, yes, you have to get across some sort of threshold, and yes, those thresholds seem to be somehow different than we're seeing, in the lymph node, for example. And I think what's happening in those tissues, often you're making cytokines like TGF-beta, for example, that push down, that change the math, that they make it harder and harder to get activated. So you're winding it down in those tissue sites for that reason. But nevertheless, effectively altering the equation of what you need to convert from sort of a tolerogenic outcome to something very stimulatory. That would be my guess of the way I would start thinking about it. But yes, I think it is a different, there is a difference that can't be directly applied.
I have a question from online, from Wayne. Insightful talk, Phil. Thank you, first of all.
Thank you.
With these immunological clocks, have inhibitors or promoters for these cellular programs been found in pathogens? For example, have homologous proteins that drive early cell death in immune cells and hinder immune response been found?
That's a great question, and I'm sure the answer is true, that there is, and I should be able to just pull out 5 examples, but I just can't off the top of my head. I know there's many examples in viruses that manipulate the cell death pathway, manipulate the antigen processing pathway, manipulate the co-stimulation pathway, but I just can't give you specific examples. But yes, I think the answer is clear. And going to the point, which I think you're really pulling out there, is that this is always a battle and a race, and there's lots of evolution on both sides. And that the history of what we see in our immune system, everything all at once, the dream is that we could read it backwards and understand that history and how it accumulated over time, and read out our infectious history as a species.
Hi, I've got a question. So I wanted to know, how might timers be implicated in immune dysfunction, and is it possible for any of the fate controlling proteins such as MYC to be considered as future therapeutic targets?
Great question. The interesting thing about, and we're working a lot on this with the Vanessa Bryant lab, is that sensing those timers, the sensing and the connection to the timers, even if you put them under very controlled conditions and look at different individuals, we find differences. And of course, when you think about it, each of those timers is summarizing the effective operation of thousands of gene products and maybe tens or hundreds of thousands of gene sequences regulating those gene products. And so, and the other point to make is that we know from our modeling that small effects, a small effect in the division time and a small effect on the destiny time, work in synergy to increase or decrease the effective outcome. So yes, we're very interested in the idea of coding the timers and the variation between people and relating it back to features of deficiency or autoimmune susceptibility, for example.
Gluten free? I feel like yes. Right? Milk and soy, peanuts and tree nuts.
There we go.
Are we insured if you take someone's eye out, is it?
I don't know, when Andrew's left.
How many people in the audience came here with the view that immunology is immensely complex? It seems to be our reputation. I'm just curious. There's a few hands, quite a lot of hands. Great. How many of you think now it might be a little less complex than when you came in? Oh, good. Couple.
You've got 22 weeks, don't worry about it.
Great. Can we give Phil another round of applause for today's seminar?